Hello, everyone. Today, we're going to discuss the answer of the UT1. So first, please look at question 1. So compare image A and image B. You will find that image A is brighter than image B. So to correct the image, so you need to adjust the microscope to make the image dimmer. Okay, so what can you do? So 6 is the things that you need to do. You need to adjust the iris diaphragm in order to let less light to enter the microscope. Okay, so it makes the image dimmer. So this is the first thing you need to do. Secondly, you will find that image A is not clear enough. Because it's not in focus. So to get focus, you need to adjust the fine adjustment knob. Okay, so 4 and 6 is the correct answer. Many of you will include C, cross adjustment knob, in your answer, which is wrong because cross adjustment knob is for large scale adjustment. Okay, you see, compare with image A and image B. You only need fine adjustment to make it clear enough. All right, so 3 is not needed. All right, for question B. So there are 2 criteria. First, the student wants to observe the cell again, but with a higher power. Furthermore, he only wants to observe cell P. Okay, so in order to observe cell P, okay, let us look at image P first. Okay, so what he need to do is he need to adjust cell P to the center of the view. Okay, so you need to move the diagram downward and leftward. Okay, however, the image that you observe from microscope is invert, right? So say, for example, if the object is P, when you look into the microscope, you will see an image like this. Okay, the image is invert. So go back here. If you want to move the cell P to center, you need to move it downward and leftward. Then to do this, you need to move the stage upward and rightward. So choice 2 is the first thing you need to do. Then secondly, because the student want to observe selfie in more detail, so it need to increase the magnification power. So choice 1 is a necessary choice. That every time when you switch the objective to change the magnification power, you need to find the focus again. So choice 4 is also necessary. So the correct answer should be 1, 2, and 4. Am I right? For question 1C, okay, organelle X contain a green pigment. Okay, first, organelle X is chloroplast. And the green pigment is the chlorophyll. All right. Many of you will write something like that. First, green pigment carries out photosynthesis, or chlorophyll is the site for photosynthesis. Okay, both of them are wrong concept. The correct relationship of green pigment and organelle X, which is chloroplast, is that, remember, chlorophyll is not the organelle. So X is not chlorophyll. Chlorophyll is only a pigment, sexual, which can be found in chloroplast. Okay, then what is the use of chlorophyll in chloroplast? So chlorophyll is a green pigment that absorbs light. Okay, because photosynthesis need light, so chlorophyll absorbs light. For organelle X, which is chloroplast, to carry out photosynthesis. So this is a correct relationship of chlorophyll and chloroplast. Chloroplast is the site for photosynthesis, but not chlorophyll. But without chlorophyll to absorb light, chloroplast cannot carry out photosynthesis. Okay, then what will be the third point? Then you need to tell us what is the aim of photosynthesis. Okay, photosynthesis is to produce food, or you can say to produce glucose. All right, then you can get 3 points. And now we move on to question 2. So if you do worksheet 1 seriously, okay, then you must be able to answer this question. Excretion means to remove metabolic waste, right? Metabolic waste means the waste produced by the chemical reaction in body cells. Okay, so when your body cells carry out different chemical reaction, some waste products, some products that we do not need, will be produced. Then to remove all this waste, it belongs to excretion. So remember, removal of feces, 粪便, is not a kind of excretion, because feces is a kind of undigested food. Undigested food is not produced by your body. Okay, you eat the food, some food substances cannot be digested and you remove it. It is not produced by your body. So choice 2 is not the correct answer. And the correct answer is urination and exhalation. In exhalation, you remove CO2, carbon dioxide. Carbon dioxide is produced by respiration. Respiration is a kind of chemical reaction in your body. Okay, so removal of CO2 is a kind of excretion. So the correct answer should be 1 and 3. For question 3. 嗱呢題錯咧，我就覺得離譜嘅。點解咧？因為我上堂講 worksheet 1嘅時候咧，其實都好重點講過嘅。Irritability is the ability of an organism to react to stimuli. Okay, if you say changes in the environment will also be accepted. However, if you just say react to environment, then I will give you only half a mark. For question 4, okay, label A, B, and C. Okay, C is obvious, the nucleus, okay? But some of you wrongly label it as vacuole or cytoplasm. Remember, C is nucleus. 最容易 label 係呢個㗎啦，其實。And then A. So this is structure A under higher magnification. You can see some black dot on organelle A. Okay, it is obvious, rough endoplasmic reticulum. So because this question asks you to name the structure, so short form is not accept. Rough endoplasmic reticulum. 嗱，再講多次，我都唔明點解我講咗幾次啊。其實做功課嘅時候同樣出現呢個錯誤嘅，好多同學寫咩 rough endoplasmic reticulum， OK？ It should be reticulum. And remember, every time when I ask you to name something, don't give me short form. Don't say rough ER. For organelle B, okay, B is rod shaped. So B should be mitochondrion. Plural form, mitochondria. So both mitochondrion and mitochondria are accepted. So if you are not able to recognize the structure, the organelle, then you will lose marks when you answer question 2. Okay, so it's very important for you to label different cell structures. So it asks you how structure A, B, and C work together to produce antibodies. And the hint is antibody is a protein. So to answer this question, okay, there is 2 main concepts. First, you need to tell what is the role of each organelle in producing antibody, which is a kind of protein. Secondly, you need to tell how ABC work together. So work together is also important. If you just state the function of 3 organelles separately, then you will only get a part of, you will get only 6 marks, okay? The final marks is for their relationship, okay? So pay attention of the style of your answer, okay? First, I will talk about nucleus first, because to produce protein, nucleus is the first organelle that you need to mention, because nucleus, which is structure C, carries DNA. Then, as we have mentioned in lesson, DNA carry genetic information. And this genetic information is for the production of antibodies. Okay, so I would like you to tell me not just DNA carry genetic information, but you need to tell me the genetic information is for production of antibodies. Then this is a very important answering skills. So if you say it carries genetic information, then you have 1 mark. If you say the information is for production of antibody, then you will get another mark. Then the second important organelle is mitochondria. Okay. Then how do mitochondria contribute to the production of antibody? Okay. Then just simply tell me what is the function of mitochondria. Okay. Mitochondria is the site of respiration. If you are able to tell me this, then you will get 1 mark. Then second point, you need to tell me how does mitochondria help the production of antibody. So all of you know that respiration release energy from food so that it supports different metabolic activity. Without the energy, different metabolic activity will stop. So mitochondria is the site of respiration. So it releases energy from food or glucose to provide energy for production of antibodies. Okay, so as I emphasize again, relationship of different organelles is important. Okay, third, which is the rough ER. So rough ER is the site for protein synthesis because it has ribosome. Okay, and antibodies is a kind of protein. So antibody is produced in the ribosome on rough ER. Okay, so for rough ER, sorry, which is A. First, you need to tell me rough ER or structure A contain ribosome. Okay, which are the site of protein synthesis. Then, in this point, you need to tell me antibody is a kind of protein. Otherwise, you will get no marks. So antibodies are proteins. All right. So if you can answer all these points, then each point will worth 1 mark. If you are able to include the linkage between different organelles, okay, then you will get another mark. So say, for example, mitochondria provide energy for rough ER to produce protein, then this is the linkage. All right. Another example, structure C, which is nucleus. They provide information for rough ER to produce antibodies. Then this is also another linkage. Okay. So in your correction, I would like you to write the answer yourself. Don't just copy my answer. I would like you to think about how do you add the linkage between each organelle. All right. 咁所以咧，嗱對我會 check 你嘅答案嘅時候咧，咁我希望睇到嘅咧係大家除咗將嗰個 function 好分散咁寫出嚟之外咧，我唔想咁樣，我想你睇下點樣樣將1、2、3呢3樣嘢 link 埋一齊啊，得唔得？咁呢就靠你啦。Right, for question B. The antibodies produced are packed into structure D for secretion. Okay, if you look at your notes, you will find that an organelle, sometimes they store useful substances, sometimes they store waste, which is vacuole. So structure D is vacuole. In animal cells, you can find many small vacuole. You can see here, here is vacuole. These are vacuole. Okay, all of these are vacuole. And the vacuole in animal cell is usually small. Question C, calculate the actual length of PQ. Show your working. So to do this question, of course, you need a ruler. So if you had a ruler, then measure the actual length of PQ. Sorry, not actual length, the length of PQ on your test paper. You will find that the length of PQ is equal to 6.7 cm. Then what is the actual length? Then, after measuring the length of PQ on your test paper, then you need to measure the length of the scale. Okay, so it should be 1 cm. Okay, so the concept here is, so if you have learned similar triangles, if two triangles are similar, then the ratio of AB to A1B1 should be the same as the ratio of BC to B1C1, right? So similar concept. So this is the actual length of PQ. on your test, which is 6.7 centimeters. Okay, about the scale bar. The actual length is 1.2 micrometer. And the length of this scale bar on your test paper is 1 centimeters. So their ratio should be the same. Okay, so you can find the actual length of PQ by using this equation. But to do this, you need to convert all the units to be the same. Okay, in this case, we will use micrometer. Okay, here, given that 1 micrometer is equal to 0.0001 centimeters. So 1, 2, 3, 4. 1, 2, 3, 4. All right. For question D, okay, it asks you to find out the magnification. I've already told you the method when we do the worksheet for microscopic diagram for microscope. Okay, magnification means how much, how many times it enlarge. So very easy. The concept is you need to use the length on the diagram, okay, divided by the actual length. Then you know how much it enlarged. Also, remember when you do the calculation, you need to convert all the units be the same. Okay, for this one, I'm not going to give you the answer. Okay, so do it yourself by using this method. For question E, very easy. Differences between bacterial cells and blood cells shown. White blood cell. Okay. Bacterial cell is a kind of prokaryotic cell. And white blood cell is a kind of eukaryotic cell. Okay, so actually this question just want you to recall what are the differences between prokaryotic cell and eukaryotic cell. Okay, so I'm not going to give you the answer. Just go back your notes, find out the answer. All right, do the correction yourself. Question 5. So you see here, all cell has a red pigment inside, but the cell Y do not have red pigment. Why? Okay. 咁有啲同學咧就話哦，因為 Y 本身冇咯。喂，咁同一個 structure， 同一個 tissue， 冇理由有啲有，有啲冇㗎，係咪 ？So the only correct explanation is that when you prepare the sample, if you still remember, you need to use a forceps to take the thin or to tears the thin layer of epidermis out. Okay, so and you know that the red pigment is found inside the membrane. So when you peel the epidermis out, the cell membrane of cell Y is broken. So the red pigment leak out, right? So cell Y is damaged. The cell membrane of cell Y is damaged. So you cannot find red pigment inside. So when tearing the epidermis, the cell membrane of cell P is broken. All right, so the red pigment leak out. You write it yourself. So for this question, there is one bonus mark. If you are able to tell cell membrane is selectively permeable, then you will get an extra mark. This point is very important because cell membrane is selectively permeable. So if it is not broken, the red pigment cannot leak out. So this point also worth one extra mark. For question B, structure K remains intact with reference to its chemical composition. Okay, so one of the keywords of this question is chemical composition. Explain why structure K can remain intact. When you peel the epidermis, epidermis 即係表皮啊 ，epidermis, cell membrane is broken, but K, which is cell wall, is not broken. Why? 咁其實都提示到好出面㗎啦，佢叫你講埋 with reference to its chemical composition。咁所以你一定要講佢嘅 chemical composition 啊。So first, K is made up of cellulose. So K is rigid and thick. So when you tease it, okay, it's not broken, okay? Okay, and cell C, sorry, not cell C, question C. Even though structure K remains intact, it cannot prevent the incident in A. 即係佢問你咩啊？喂，個 cell membrane 爛咗啫，個 cell wall 冇爛喎，點解佢阻唔到啲 pigment 流出嚟啊？咁呢題啊反而答得幾好嘅。So because not like cell membrane is selectively permeable. So it can stop the pigment from leaking out. But cell wall is freely permeable or fully permeable. So even cell wall is intact, the pigment can still leak out. All right, so this is the answer.